I'm going to try to stay out of the way by slides. Title of my talk that you've probably seen if you look at the descriptions is, Are Aliens Hiding Behind the Cracker Barrel in Nebraska? And I'm immediately going to tell you the answer to that. And it is no. So I'm very sorry if you're disappointed and you feel clickbaited, but you're already here. So I guess you might as well stay and listen to the rest of the talk. But there is something behind Cracker Barrel. And it can help us understand extraterrestrial life, potentially. So not quite aliens, but it's not totally irrelevant. So hope you don't feel scammed, but hopefully you're not too disappointed by the end. So when we try to find extraterrestrial life, we often think about Mars first. And there are a couple of reasons for that. First is that Mars is close by and space travel is really expensive. So it's a convenience factor there. The second thing is that Mars is kind of often called Earth's sister planet. It is the most Earth-like planet that's in our solar system. And despite it being the closest to Earth, Mars will kill you. Mars takes no prisoners. If you want to be semantic about it, technically the sun will kill you. The sun is the problem. It's not Mars itself. But for all intents and purposes, Mars will kill you. And the reason for this is that on Earth, we have a magnetic field, which is really neat. And we have an atmosphere. And both of these things kind of deflect a lot of the solar radiation. But on Mars, that is not the case. And so any solar radiation pretty much ends up at the surface of the planet. And that's a problem for life. And so we can think about the most radiation-resistant organism we know, which is Deinococcus radiodurans. You would die 3,000 times before this organism dies once at the same level of radiation. So crazy amounts of radiation it can tolerate. And it even has a Guinness World Record for the world's toughest bacterium. I don't know who decided that or what, credentials they have to decide what bacteria gets world records, but it has one. And so even that organism will die within 7 days on Mars. And so that's a problem. And so you might be saying, okay, well, do we give up on Mars? Is there no life there? If it's not possible. But there's the underground. And this cool thing about the underground is that UV doesn't go there. And so if life were underground, it could be totally fine. But there's one caveat, and it's that underground, there's no CO2. Well, if there is, it's very limited. But any organism that we think would be there would be self-sufficient and able to fix its own carbon. And so it would need that source of CO2. And so there's something in the subsurface that's not CO2 that maybe could be CO2, and that's calcium carbonate. And so calcium carbonate, also known as chalk, limestone, it's the active ingredient in antacid tablets. So whatever is most relatable to you, you can go with that analogy. But I'm trying to kind of figure out if this calcium carbonate could be a viable substitute carbon source for microbes in the subsurface that would use CO2. And so microbes in the subsurface that would use CO2 is a pretty broad category. So I look specifically at methanogens, which are archaea, and these archaea make methane. So methanogens are generating methane. And we kind of have to narrow it down a little bit to have a direction for our research. So basically what I'm trying to address, I have these methanogens, well, I'm going to have them. I want to know if they can use chalk instead of CO2, and as a proxy to understand if this could be happening in the Martian subsurface. So we need methanogens. Where are we going to find them? And this is our brief stint where Cracker Barrel plays a role. And so it's not Cracker Barrel per se. We're not going in there and collecting samples. And it's not quite behind it. We're not like at dumpster level or parking lot, but even further behind, a particular Cracker Barrel here in Nebraska is a saline wetland. And so this wetland was the source of our soil sample that we used to kind of enrich for a methanogen. And so we did that. We took soil. We put it in these little bottles, sometimes big bottles, but full of a really minimal medium. And the white is chalk, calcium carbonate. And we just let them hang out. and we transfer them once in a while, trying to kind of weed out the hitchhikers that aren't actually using the carbonate mineral. And we did eventually get a methanogen, which is good. That was our goal. The problem when we did DNA sequencing to see who was there is that we also have a bunch of not methanogens. Not ideal. I want to study specifically the methanogen and what they're doing. And so having all these other organisms is like if I was playing a song for you and I wanted you to write down the lyrics and you had never heard this song before. But I also turned on 5 TVs at full volume and they're all playing different things. And your upstairs neighbors just got their kids a pogo stick and a drum set. And then there's also a plane outside that's trying to break the sound barrier, all while you're trying to transcribe these lyrics for me. So you could try, but it's going to be a lot more difficult than it would be as if everything was quiet and you could just listen to just the song and just write that down. And so that's kind of what we're trying to do is just have the methanogen, just have the carbonate, just figure out what's going on strictly in a simpler system. And in order to do that, we have to isolate the methanogen from the other organisms. And so this is where I start to hate science. And this is where the story begins, where things get very frustrating and very difficult. And so it's easier said than done to try and pull one organism out from a group. And there's a lot of approaches that you can take to do this. And I'm going to talk about a couple of them. So here's our community that we started with. Again, we just want these yellow rods, which we're pretending are a methanogen. And so microbes have different nutrient requirements. So one thing that you can do is kind of manipulate the nutrients that you provide and sort of weed out organisms that can't use those nutrients. And so it's a way to kind of select just for certain organisms. And if you're lucky, it works. And if you're me, you get rid of like one organism. So it kind of works. That's progress. Unfortunately, it's not really going to help us too much. So we have to try another approach. Another approach would be to kind of dilute your culture to the point where you have very, very few cells, and those very, very few cells will grow up and then populate your culture. So it's called dilution to extinction. You're basically trying to get rid of everything you don't want in hopes that what you're left behind with is what you did want. When we do that, I get rid of my methanogen, and I kind of needed that. So then I'm left with some cells I don't want, and the stuff that I do want is totally gone. So that's okay. Fortunately, we have the cultures to go back to, because if we didn't, that would be bad. We'd have to go back to the wetland, to the Cracker Barrel. And so another final approach that I'm going to talk about is using antibiotics. And this is especially helpful because we're trying to study an archaeon. And antibiotics usually target specific physiological features. And a lot of the features in bacteria that antibiotics target are not present in archaea, so they should be unaffected. And so I introduced a antibiotic that shouldn't, keyword shouldn't affect my methanogen, and I killed completely everything that was ever in there. So we back up again. We're like, no, thank you. We need those things. I can't study nothing. And we try a different antibiotic. And then we kill everything again. And so basically I spent 4 years doing various combinations and permutations of this process, all these things and more, trying to isolate this methanogen, and it doesn't work. And I don't have kids, but I imagine this is kind of what having kids is like. And it's that you provide them everything that they could possibly need, and you set them up for success, and they don't appreciate it. At least that's what I've heard. It's like having kids. And the ironic part is that in the wild, archaea are super, super resistant to so much. They can be found at the ocean floor, Yellowstone, anywhere. But once you bring them into the lab and give them everything they need, they don't want it. And so why am I still doing this, basically? After 4 years of failing to do this, why am I still doing it? And well, first, the little things are so much more enjoyable. And the little successes are so great. Like, yeah, I didn't isolate my methanogen, but I didn't kill it this time. And second, we still learn something throughout this process. Even though I didn't necessarily get from point A to point B to my goal, I still was able to learn something about these organisms through the trials and tribulations of trying to isolate. That is it.